first class. So I and my friend Tejaswini will introduce you to the basic topics of microbiology which are required in when you work in the laboratory. You need to know some basics, and we will discuss you. We will discuss you that today. And just wait. First, I want to thank you all, and I want to thank Biovigan to provide us this opportunity to present this session today with you. So this is an interactive session, guys. We are not your professors. We are just your friends. So you can ask your doubts. Even if it's a very simple doubt, you can clear that with us. So before proceeding, I want to know if you guys are familiar with the word microorganisms. If someone can tell me what are microorganisms. So I can't see your faces basically. So I need to know that if you know what are microorganisms, please say, yeah. So someone has raised their hand, Sayed Tariq. So you can answer like what are microorganisms.
Guys, don't hesitate. It's okay. If you give wrong answers, also, we are here to help you up. So don't freak out. Say whatever you feel. What are microorganisms?
Okay, I think I should continue.
Hello,
Yeah, yeah, you're audible.
am I audible?
Yes, yes.
Basically, my name is Dute. So the question is like, what is microorganism? So basically, what I know is it's basically an organism which is of microscopic size, and it may exist in a single cell or as a colony cells. Yeah,
Yeah, so we cannot see them with our naked eyes. That is correct.
yeah.
Yeah, so they are found everywhere. Okay, like microorganisms are ubiquitous. That is, they are found everywhere in soil, in air, in water. Like without microorganisms, the ecosystem can't work. As humans, we can't work without that because our gut, our skin, everything has microorganisms. Okay, so the world is incomplete or the planet is incomplete without microorganisms. So, as microbiologists, what our role is to preserve or to isolate this bacteria and make use out of it to produce enzymes or for in genetic engineering or like there are innumerable examples of what we can do with microorganisms, as we all know. So first, our aim will be to obtain a pure culture from mixed colonies or mixed cultures. Okay. So first, I will tell you like what are the basic laboratory apparatus you can find in a lab when you are working. You will come across this autoclave and incinerator, Bunsen burner, and these basic things. So we will be talking about media. We'll be talking about how to isolate and preserve these colonies. So in that, broth, semi-solid, and solid medias are there. So we'll be talking about everything in our upcoming slides. So this is a pictorial format of what you can find in the laboratory, like a hot air oven, incubator, microscope. I hope that you all know what users this all have, basically. So if you have any doubt, you can ask me. Like if you don't know anything, you can ask us. So before proceeding to obtain a pure culture, we have to know what sterilization is, because we have to sterilize everything we use. We when we keep things in the laminar airflow, we have to sterilize them before use, so that we don't get any contamination. Okay. So sterilization is done to kill everything, like kill all the microorganisms present in the petri plates, in the media, and whatever you are using. So it can be of various, it can be done in various ways. Okay, so first is autoclave. This is the most used method or the most used technique in for sterilization. It works in the principle of pressure cooker. We all have used it. We have at least seen it in our lives in the kitchen. So it works in a similar way, and it kills all the microorganisms because it works in 121 degrees Celsius at 15 psi for 15 minutes. We have to keep it in 121 so that everything dies. I mean, kills everything. For example, it will kill the bacteria, viruses, fungi, and everything, and also the endospores, because the endospores are very resistant to heat, as you all know. So we have to kill those also, because if they get the favorable environment, they will grow, right? So we don't want that. We don't want any contamination. So we have to kill those as well. So autoclave is one way of doing that. The next is filtration. When autoclaving is not permeable for some media or antibiotics, serum, etc., so we can use this filtration. It filters out the, it retains the microorganisms, and the filter we get is the, uh, we cannot say it's sterilized like 100%, but yeah, it kills almost everything. It inactivates. It is done with a filter which is made up of cellulose acetate or polycarbonate. Okay. The next is chemical sterilization, which is done with the use of ethylene oxide gas, hydrogen peroxide, and glutaraldehyde. It inactivates or kills the microorganisms which are present. And this is also done when autoclaving is not possible. For example, when you're using plastic petri plates, it's not possible to autoclave those petri plates, right? Those are disposable ones. So we have to first sterilize them with chemicals, and then we can use it. And other things are electronic devices and certain other medical instruments which you can't autoclave. Like moist is not possible for those. So we go for this chemical sterilization. The other methods are dry heat sterilization, which is done at 160 to 180, so which is like very high, and it can kill all the spores and bacteria and everything. UV sterilization, which we use in laminar airflow. Generally, we switch on UV for 15 minutes to kill or inactivate all the microorganisms which are present in the laminar hood, so that we can maintain that pure, the sterility, So, and the next is the flame sterilization, which we do to kill all the microorganisms. Like we flame the loop, we flame the L rods and everything to kill all the microorganisms before we proceed to streak or spread in our plate. So this is what I explained. This is at a glance. You can see here that heat can be of moist and dry, and filtration, chemical, and radiation. Okay, so this is everything at a glance. So before proceeding to culture media, if you have any doubts regarding sterilization, you can make sure to ask us, or I'll be starting with culture media. Okay, I think I should go on with culture media. So what happens is, what is a media? Like, yeah, different bacteria have different nutritional requirements, as we all know. So we have to provide them with those nutritions to, in order to make them grow. Okay, so this media will provide them with the sufficient nutrition and whatever requirements they have, so that they can grow properly. One of the basic media which we use in laboratory is nutrient agar, which supports the growth of all kinds of microorganisms. It can be bacteria, fungi. It supports everything. That is the basic or the first media which we use in laboratory. So now the media can be a liquid, solid, or semi-solid. Okay, as you can see in the picture, the first is a petri plate, which is solid. The second one is liquid or the broth medium, and the third one is semi-solid. So why do we use these three? Have different uniqueness, like for which we use it. So the solid is of course for plates, and we can also use like slants and everything, which I'm going to talk ahead. And liquid medium is the broth medium, in which when we want a higher load of the microorganisms in our broth or liquid culture, we can go for this broth medium. And the last one is semi-solid, which we use for several tests, like bacterial motility, etc., like gaseous exchange between the environment and the bacteria. So for that we use semi-solid medium. So now agar, we all have used agar in our laboratories, but we don't know if it provides nutritional value or it's just a solidifying agent. Like we always have such doubts when we are working, but like today we want to clear those doubts for you. So agar doesn't have any nutritional value. It just acts as a solidifying agent when you are adding in a test tube, or it may be in a test tube or a petri plate. It just helps to solidify. Okay, it is derived from seaweed, which is a complex carbohydrate, and it is just a solidifying agent. Okay, and it, like, the unique property is that it can be used for the cultivation of organisms at elevated temperatures, because it won't liquefy. Like, even if we keep it at elevated temperatures, after it gets solidified, it won't liquefy. So that is a plus point for using agar. The concentration of agar we use in different I mean, it differs from lab to lab, but the standard concentration is between 1.5% to 1.8% of the media. Like, if you are preparing 100 ml, it can be around 1.5 to 1.8 grams of agar. So, okay, the right, in the right side, you can see that there's a bottle of agar which we generally use. And the applications of semi-solid medium, as I already told you, that it's used for motility, and like the for checking the oxygen levels or the growth of bacteria in the presence and like in the absence, if it's aerobic and anaerobic. So in that manner. Now we come to agar slants, deep tubes, and agar plates. So agar slants, why are these used? These are, firstly, you have to pour the media in a test tube. Okay, 10 ml, around 10 ml you can pour, and then you have to keep it in 30 to 45 degrees, like elevated from the mouth of the test tube, and then it will prepare. Then you will prepare a slant, and it can be used to preserve the culture for a longer period of time. Basically, slants are always used to preserve the culture, because there are less chances of the culture getting contaminated when we prepare slants. So you can use them for preserving. And agar deep tubes, they are again used to check the gaseous requirements. If it's aerobic, anaerobic, they help us to identify if the bacteria requires oxygen or doesn't. And the agar plates, they are used to isolate the colonies, as we all know. Okay, now I'll be talking about the classification of media. So, media, different bacteria have different nutritional requirements. So, we have to provide that, or else our bacteria, I mean, they won't grow. You have to provide them with the right nutrition in order to grow them. So, the media can be classified in three parameters. The first one is composition, the second one is purpose, and the third one is physical state. Okay, the first one, composition, like when we know what we are adding in the media, that like in the proper, systematic way, if we know what we are putting in the media, that is chemically defined media. But generally we use complex media or chemically undefined, because we use yeast extract and beef extract, etc., and we don't know that like what is it made up of. We just get a bottle of that and we use that, so it's chemically undefined. So, based on purpose, it can be nutrient selective, differential, or enriched. I already told you about the nutrient one, because it supports everything, like the growth of fungi, bacteria, everything. And the selective, differential, enriched, I'll be talking in a minute. Before that, based on physical state, solid, liquid, and semi solid, I already explained you. The solid plates, broth, and the semi solid like media. Now we move on to selective media. So, what are selective media? As the name suggests, they select bacteria. They select what can grow and what cannot. They inhibit some microorganisms, but they enhance or they support the growth of other. So, in the picture, as you can see, there's nutrient agar in the first one, and in the second one, it's phenylethyl alcohol agar. So here you can easily distinguish. On the right side, you can easily distinguish that E. coli is very much, transparent kind of colonies they have given, and yeah, the Staphylococcus aureus, they have given a whitish colonies when grown in phenylethyl alcohol agar. So it supports the growth of gram-positive organisms only. So that is why the S. aureus is growing and the E. coli is growing, but you can easily distinguish between them. But that is not in the case of nutrient agar. Everything looks same. There might be chances that you are getting other colonies as well, along with E. coli, S. aureus, and everything. So we go for the selective media once we isolate it in nutrient agar. When we want to obtain the pure culture, we have to check that if we are in the right track. Other examples are crystal violet agar and 7.5% sodium chloride agar. So in the crystal violet agar, it is for gram negative. It's just the opposite of phenylethyl alcohol agar. It's for gram negative organisms, and the 7.5% sodium chloride agar is for halophilic, as the name suggests. Sodium chloride is a salt, and the salt-loving bacteria will be able to grow in these media. So now we move on to differential media. So, as the name suggests, again, that differential means you can distinguish between two types of colonies in these plates. But again, like these are all interrelated terms. Okay, selective media, differential media. It's not like differential. It's only differential. It might be selective as well as differential, which I'll be explaining in a detail. So the example for this is mannitol salt agar. In this, Staphylococcus species can grow because they are salt-loving bacteria, and only high salt concentration bacteria will be able to grow in MSA. So they will give yellow colonies because they are able to ferment mannitol and produce yellow colonies. But the other colonies will appear pink. So now, like when we club these two terms together, selective differential media, we get something where we can select a bacteria, like it inhibits some and supports some type of microorganisms, as well as it helps us to differentiate two types of colonies. So here, one example is McConkey agar, which we use frequently in our laboratory. So here the selective component is because it has bile salts and crystal violet, which inhibits the growth of gram positive organisms but allows the growth of gram negative bacteria. Okay, and the differential property it has is the negative organisms will produce, which can ferment lactose. I'll explain that with a picture. So here, as you can see in the left side, E. coli has grown and it has given pink color in the plate because it is fermenting lactose first and it is producing acid after fermentation of lactose. And then what is happening is the precipitation of bile acids is happening and it is giving that red color because of the absorption of the neutral red which is present in the media. And in the right side, these are non-fermenters, so it won't give any pink colonies or anything. It will just turn the media yellow, so you'll be able to distinguish between them very easily. So the last one is the enriched media. So as we know that some microorganisms have special requirements, okay? So these are for such organisms which are also known as fastidious microorganisms. So they want high nutrition. For that, we add blood, serum, or yeast extract, etc., to cultivate these fastidious organisms. So here, fastidious means it is, difficult to isolate these organisms in the laboratory. So here, the blood which is used is generally sheep blood in for blood agar, and it is done for streptococcus as well as Neisseria. It can be done, and yeah, in the blood agar, you can see that different types of hemolysis has taken place. So hemolysis only takes place when the bacteria can secrete hemolysin, which in turn can cause hemolysis. Okay, so here, like what happens is Streptococcus can either produce hemolysin or cannot. So in the first one, as you can see, the gamma hemolysis, in that no lysis of red blood cells has taken place. So the bacteria, the plate is not, the color has not changed. The bacteria has just grown on the plate, but no lysis of red blood cells has taken place. In the second one, alpha hemolysis, it's an incomplete or partial lysis of red blood cells. So now, when the red blood cells are lysed, they produce, they give out hemoglobin in the media. Because as we all know that red blood cells carry oxygen with the help of hemoglobin. So that comes out in the media and the bacteria reduces this hemoglobin to methemoglobin, which in turn produces the green color, the halo around the bacterial growth, as you can see in the second picture. This is known as alpha hemolysis. Now, the third one is beta hemolysis, when complete destruction of red blood cells is occurring and the complete use of hemoglobin is also done. So, then you will get this clear zone around the colonies, and you will be able to distinguish that okay, this is beta hemolysis. And when it is beta hemolysis, you can also know that okay, my bacteria is producing streptolysin O and streptolysin S, because these are required for destructing of the red blood cells and use of hemoglobin completely. So these are done to like differentiate that okay, this is gamma, this is alpha, and if my bacteria has those and enzymes to do this. So I hope that these things are clear. Like if you have any doubt, you can ask us, or I like to hand over this presentation to my friend Tejaswini, who will talk about how to obtain a pure culture from a mixed culture. Thank you so much. If you have any doubts, please don't hesitate to ask them.
No doubts?
Okay.
Okay. Thank you, Saurabh, for handing over the platform to me.